Good afternoon. My name is Pietro Miozzo. Today I will present my research on the role of the lupus-associated protein LIPW in response to lymphocytic choriomeningitis virus infection. This exciting work was carried out in Dr. Eric Peterson's lab at the University of Minnesota with support from the Lupus Foundation Minnesota's Summer Research Fellowship. Systemic lupus erythematosus is an autoimmune disease that affects many tissues throughout the body, including the skin, kidneys, and joints. The immune system attacks the body's cells, and inflammation and tissue damage ensue. The cause of SLE is unknown, but white blood cells, like dendritic cells, are involved. Dendritic cells are a key part of the immune system that could direct tissue damage in lupus. We also know that there are many genetic factors and environmental triggers. One of the genes implicated in lupus is PTPN22. In humans, the gene PTPN22 codes for the protein LIP. People with SLE may have a variant called LIPW. LIPW has been strongly associated with lupus, and the variation comes from a change in the DNA sequence that results in a different building block in the protein. What does LIP do? A subset of white blood cells, these dendritic cells, can detect infection stimuli through a receptor. When the pathogens are detected, type 1 interferon is produced. Type 1 interferon is a chemical messenger involved in the activation of white blood cells and fighting infection. For this to happen, LIP is required. The major variant, LIP-R, helps produce large quantities of type 1 interferon. However, the lupus-associated variant, LIPW, has been shown to produce less type 1 interferon. So, we say that LIPW exhibits a loss of function. This is an important direction for lupus research, as altered levels of type 1 interferon have been associated with the disease. Given this protein's important place in fighting infection, I set out to examine the role that LIPW plays in response to lymphocytic choriomeningitis virus infection. LCMV is a widely used virus in these kinds of experiments. How do I measure a response? I can look at a group of white blood cells called dendritic cells and measure their production of type 1 interferon and the presence of activation markers. We need to consider these cells in lupus research because they are the largest producers of type 1 interferon. The process by which these activation markers are presented is dependent on type 1 interferon. To detect proteins on the surface of cells or within them, we can use a technique called flow cytometry. Before we go any further into my experiment, I think it's important to have an idea of what flow cytometry is, especially given that it is the cornerstone of modern immunology and lupus research. Cells have proteins on their surface, like activation markers, or within them, like type 1 interferon. These proteins can be thought of as a lock. To detect them, I can stain them with antibodies that have been coupled with a fluorescent molecule. This is like a key. An antibody fits only its own protein, like a key will only fit its own lock. When the machine detects the colored molecules, I know the protein that is being displayed. The flow cytometer's readout looks like the graphs on the bottom, where each dot represents a cell, and the graphs show the level of one protein compared to the level of another. Having posed my question, I then set out to consider possible outcomes. Based on previous work by the Peterson lab and what I've already shown you, I hypothesized that LIPW would cause decreased levels of type 1 interferon and a decreased display of activation markers. Therefore, LIPW would have a reduced response to viral infection. To test this hypothesis, I used mice with modifications of the PTPN22 gene. This is the LIP-producing gene. In our research, we use mice for several reasons. We have to be able to control genetic backgrounds to ensure homogeneity in our experiments, and mice help simplify this. Furthermore, small animals provide us with precise tools to that we can use to modify our conditions and ask questions about different genotypes. Finally, we have direct access to tissues and processes that we might not otherwise be able to reach in humans. Using the animal model, I was able to carry out my experiment by comparing different variations of the PTPN22 gene that produces LIP. As a negative control, I used uninfected normal mice. These are mice that have an intact copy of the mouse LIP-producing gene and are not infected. To examine responses to LCMV infection, I compared normal mice, mice without the LIP-producing gene instead of the mouse gene, mice with the human LIP-R-producing gene instead of the mouse gene, and finally mice with the lupus-associated LIP-W variant human gene instead of the mouse gene. These last two mice are called transgenic, meaning that they have a gene inserted, in this case a human gene. The infected mice were inoculated with LCMV, and after 24 hours I collected spleen. I analyzed the spleen for type 1 interferon and activation markers by using flow cytometry. The activation of dendritic cells can be measured by quantifying the presentation of two surface markers, CD86 and CD40. The lip-null mice show severely impaired production of these activation markers compared to the normal mouse. The negative control is set at 1 to compare the infected mice to a base level of activation markers within the uninfected mouse. These data demonstrate that type 1 interferon-dependent events are not as efficient in mice that lack LIP. This means that LIP is required for dendritic cell activation, shown by lowered levels of CD86 and CD40 within lip-null mice. Now we can compare mice with a lupus-associated LIP-W and the human LIP-R found more commonly. Looking at the same activation markers CD86 and CD40, we see that LIP-R is more efficient at activating dendritic cells than LIP-W. This fits with the idea that LIP-W is a loss-of-function allele. Given that these activation markers are dependent on type 1 interferon, these data suggest that LIP-W is in fact less efficient at producing type 1 interferon than LIP-R. To demonstrate the role of LIP in type 1 interferon production, we can compare a LIP-null mouse, which has been infected, to a normal infected mouse. We see that the LIP-null mouse does not produce nearly as much type 1 interferon as the normal mouse, which has a 40% increase in interferon production over the uninfected mouse. In fact, the LIP-null is much closer to the uninfected negative control. From the data I have shown you earlier, we can see that the upregulation of activation markers correlates with the efficient production of type 1 interferon. While I have not shown here a difference in interferon production between LIP-R and LIP-W mice, the deficient activation of dendritic cells strongly suggests an impaired ability to produce type 1 interferon, as we see a pattern here between activation of LIP-null mice and normal mice and production of type 1 interferon within the two mice. Before we wrap up, I would like to provide you with some take-home points. Among the myriad genes and environmental triggers, we have focused on PTPN22, a gene that codes for LIP. LIP is involved in the production of type 1 interferon, and altered levels of this chemical messenger are found in people with systemic lupus erythematosus. I hope that if only one thing sticks from what I've shown you today, it's that LIP-W, the lupus-associated variant, results in a lowered response to viral infection. We have seen that LIP is required for the efficient production of type 1 interferon, and therefore is required for dendritic cell activation. So, LIP-W does not respond to viral infection as strongly as LIP-R, because it does not activate dendritic cells efficiently. Finally, since activation is dependent on type 1 interferon, lowered activation in LIP-W also suggests a lowered production of type 1 interferon within LipW mice. So what does this mean exactly for people with lupus? Is it just that LipW makes you more susceptible to infection? How does this relate to tissue damage? Well, it is thought that PTPN22 is involved both in the fighting of infection and in regulating the immune system. By studying the gene and its protein in one context, viral infection, we are able to understand how its function might change in another context within a disease system like lupus. The body must find a balance between mounting infection and regulating inflammation that might cause tissue damage. Before I take any questions, I would like to thank those who have helped me in this project. First and foremost, Eric Peterson has been an outstanding mentor on so many levels, preparing me for my experiments, directing me in my reading, and motivating me to always read and ask about more. Even more importantly, he has shown me how to be a scientist, and, as a physician scientist himself, has given me invaluable advice in my future aspirations for the same work. I am greatly indebted to Dr. Yaya Wang, Research Associate in the Peterson Lab, for technical guidance and help with experimental design. Also, Fernanda Shoyama, a scientist in the lab, provided me with hands-on training to carry out the various assays and handle the animals. Along with Juliet Crabtree, a graduate student in Dr. Peterson's lab, she spent many hours with me both at the flow cytometer and on the computer analyzing my data. The Peterson Lab has been a great place to do science. I have appreciated the great spirit that runs through all the undergrad members and everyone else. Without Jennifer Monroe of the Lupus Foundation, this research would not have been possible, and I would not have had this experience. I've had the privilege to observe the sterling work she does, and humbly thank everyone involved for supporting myself and the other fellows. Sarah has worked very hard during the summer to make us feel welcome in the Center for Immunology and to make sure things all run smoothly. Finally, to you, the members and supporters of the Lupus Foundation of Minnesota, I now have the opportunity to thank you in person, and I hope you can see the difference your support makes. If you're interested in hearing more about the fellowship, you can check out the blog on the Lupus Foundation of Minnesota's website, right here on the bottom of the screen. I cannot say enough how much I've found this fellowship to be useful in my future endeavors and to be such an enjoyable way to be exposed to modern immunological and lupus research. At this point, I am happy to take any questions. Thank you very much.